Okay, so this is going to be our video on buffers. This is more just a little bit of notes here to supplement so that we're ready for our lab tomorrow. So when we talk about buffers, we first need to introduce this concept of weak acids and bases. So what you're familiar with so far, we did a lot of labs and titrations and stuff with hydrochloric acid. Hydrochloric acid is not a weak acid. It is very strong. And if I write the hydrochloric acid equation, when it reacts with water, it forms the hydronium ion H3O+, and it forms the leftover Cl-. And I've only drawn that arrow going to the right because this dissociation, when the H leaves the Cl and attaches to the water, is strong. It doesn't go backwards. I mean, maybe it goes backwards so slowly that you hardly can see it at all. We can effectively assume that this reaction has no backward movement or none that's perceivable at this level. We're to contrast that with an acid like acetic acid. Here is acetic acid, and he reacts with water to form a similar H3O+ by losing his hydrogen, which leaves the acetic acid as the acetate ion, CH3COO-. However, this one does go backward. The CH3CO- plus the H3O+ will go backwards and form its original products, the CH3COOH, and back to the water. So we can actually write an equilibrium expression for this. The equilibrium constant for this would be the products, the H3O concentration, times the acetate concentration, the CH3COO, divided by the acetic acid originally. And we don't typically write water into this equation, and the reason for that is if you had a big beaker of water like this, and you put some acetic acid in there, the acetic acid is such a small percentage of the amount of particles inside of that beaker. I mean, the water is like everything, right? It's the solvent. It's ubiquitous. The acetic acid, you maybe put in a few grams of this, but there's like hundreds or sometimes thousands of grams of water. The water, we assume, is just always going to be way more concentrated than all the other species in this solution. So we don't consider water in the equilibrium constant expression for an acid, for a weak acid, just because there's so much more of it than everything else. Sometimes you'll and there's usually a special name for this. Instead of KEQ, a lot of times they'll call this the Ka, the acid dissociation equilibrium constant. So this is for acetic acid. So what that means is, inside of that beaker, if I were looking at any given time, remember when we have an equilibrium system, I'll draw the beaker really big. Whenever you have an equilibrium system, you have all of those things inside the beaker at the same time. You've got some H3O+, you've got some acetate ions, and you've got some CH3COOH's in there, and they're all floating around together, right? So I've drawn, there's obviously a lot more than this. All coexisting, and then of course the ubiquitous water molecules are in there as well. But this creates an interesting situation, because at the same time what you have floating around, you have, I'll draw in red, the acid molecules. Here are the H3O+, the hydroniums, and the acetic acid, of course, is also an acid. But you've also got a lot of these, or some of these, some acetate ions, which are like weak bases, which is why I've circled it in blue. All right, and unlike, if I go back up here to our originals, unlike here, this chloride ion couldn't push backward. I didn't draw an arrow here. That's not a thing. That chloride was not a strong enough base to make this reaction go backwards. But this acetate ion is, and we see it because there's an equilibrium. It makes that arrow long enough that it can go backward. It's fast enough acting as a base to take that hydrogen off the hydronium ion and turn back into CH3COOH. So we would actually consider this CH3CO minus a base, whereas with the chloride, not so much. It's so weak it doesn't matter, essentially. So if I get down here, I'm looking at a solution where all the things I've circled in red are acidic and can function as acids, and all the things in blue are and like the CH3COO, the acetate ion, are bases. Maybe not very strong bases, but they can act that way. So a solution like this, let's say I had some acid, some HA. If I dump some HA into this solution, there is a base right there waiting for it. And remember, bases neutralize acid, so that base can take out that acid. But now let's say I add some A minus. I try to add some A minus into this solution. I add some base into this solution. There are some acid particles in there just waiting to neutralize that base. So whenever you have a weak acid or a weak base solution, like I have here, you wind up with both acid particles and base particles in equilibrium with each other in the solution. You have created what's called a buffer. You have created a solution that has the ability to react either with acids or with bases, which is different, again, than what we're used to. We're used to strong acids and bases. A strong acid, like this HCl, can only react with a base in order to be neutralized, okay, because he only creates that H3O+. But a weak acid, which creates the H3O+ and a base at the same time, has created a solution that can either neutralize acids or neutralize bases. That's a buffer. A buffer could neutralize either one. So let's go back and talk about last week. Last week you had this guy, NaHCO3. And you dropped him into water. This is some baking soda, right? With some carbonic acid. And what you got was, once it's in water, these guys separate right through here, and you get the Na plus floats off by himself, and you get the HCO3 minus floats off by himself. Okay? And what we're really interested in is the HCO3 minus. So let's do a reaction with him. HCO3 minus. Okay? Weak acid also, but can also function as a weak base, and I'll show you how that works. If HCO3 reacts with H3O+, reacts with an acid, essentially, what happens is the H3O+ donates the hydrogen over to here, and what you get is this. You get H2O left over because he lost his H+, and then you get H2CO3. Okay? But you can do the opposite. So if you had an HCO3 minus, instead of adding an acid to him, let's say we put him with a base, a hydroxide. That's a base. Okay, what we get then is the hydroxide, remember bases take protons, the hydroxide will take that H, turn it into water, and what will be left is a CO3, a carbonate ion, a -2. So this HCO3 has the potential to act as either an acid or a base. That's kind of like, that's a buffer solution right there. And so your body uses this all the time, the carbonate ion, to control, for instance, your blood pH. If you were to suddenly eat something or drink something that was very acidic, that had a whole bunch of H3O+ ions, that would be bad for your body. Your blood cells can't function outside of a pH that's very narrow. the pH in your blood is usually between 7.3 and 7.4, around there. Maybe it's like 0.35 and 0.45. If your blood is not in that pH range, it denatures the proteins, which means it changes their shape, which means they don't function as well. And so it's very important to keep your pH at 7.3 or 7.4. Well, remember, with acids and bases, if you add a whole bunch of H3O+, you're changing the pH. You're making it very low. If you add a whole bunch of base, OH-, you're changing the pH. You're making it high. And that's bad for your blood. So what your body does is it keeps a lot of this chemical around, this HCO3-. That way, if any acid comes in, it can react with the acid, turn it back into water. That's neutral. Okay? But if any base comes in, it can react with that and turn it into water instead. So maintaining a large amount of this chemical, this bicarbonate ion, is very important for your blood. And that's the concept of a buffer. A buffer is something that's going to resist changes in pH because it has the ability to neutralize both acids and bases. And usually, a buffer then just requires something like HCO3. HCO3 was a very weak acid, or came from a weak acid. We had the carbonic acid that you used, H2CO3, in that lab, is a weak acid. And so when it dissolves here into H3O+, and then HCO3-. And so we were taking this chemical and saying that's going to be a good indicator, because it can go backwards or forwards. Just like when we had over here. Oh, I had it up here somewhere. Where did I have it? Yeah, the acetic acid, which did the exact same thing. Acetic acid was another weak acid, just like carbonic. This is our concept of a buffer. We are not going to do the math associated with buffers, so you can thank your lucky stars for that one. You will get into it next year in AP, but we are going to be playing around with this idea of buffers and pH resistance to change. So tomorrow, when you come in, we're going to be taking a solution. And you did this a little bit. Okay, you had a flask or something with water in it, and then you had another flask with water in it. This is with the last week's lab, or the cobalt chloride lab. And you added some acid. You had some HCl that you poured in, which you would expect the pH to go way down. And when you didn't have the buffer in there, it did. It went from like pH 7 down to like pH 3. That was what the acid did. Okay, big drop. Then you added the buffer, which was the bicarbonate ion. Was in here, and then you tried adding the HCl again, and it went from 7 to like 6.5 or something, which is a much smaller change. Okay? We're going to be doing this tomorrow, exploring the effects of CO2. So one of the things Mr. Kipp tried to do in the last lab when he was running out of carbonic acid is he took a beaker of water and he took a hose. So here's the hose. And basically the hose was attached to a flask that was generating CO2. So little arrows here indicate that the CO2 went up into the flask and then bubbled up through the water. And that actually is how you make carbonic acid, because the CO2 will react with the water, gain some hydrogens, and make some H2CO3 left behind. A lot of the CO2 leaves, but some of it stays. We're going to be trying to do this tomorrow. You're going to be trying to make carbonic acid inside of a beaker. And you're going to see whether or not you're successful in that, just in the pure stage, and also when you have a buffer in place already. So if that water had had a buffer in it, I would have had presumably a lot harder time with it. Okay, this is the behavior of buffers. If there's a buffer in the solution already, you see a much smaller change in pH. Okay.